So hi everybody, it's a great pleasure to be here for this BIOD25 Genomics class. And today's lecture is going to be about transcriptomics. But before we get started on transcriptomics, I'd like to make a couple of comments and general announcements about the class. Today is not very dense, a little bit less dense than last week, so we have time for a bit of discussion. So the first item on my list is Pcr Primers. So as you have noticed, as some of you have noticed, I have made a video on YouTube to explain to you how to design primers. It's because last year there was approximately 1/3 of the people who really had trouble with designing PCR primers, and that's really the most basic task you can think of in terms of molecular biology. So I made this YouTube video hoping that you guys would watch it and be able to solve the problems. So this year there still are approximately 1/3 of people who get them wrong. So my advice is watch the video again and again. If you got both primers wrong at the quiz, then you didn't get it. Watch the videos as many times as needed until you get it. And so as long as we have a high number of people who cannot get the primers right, I will keep every week asking you questions about PCR primers. So the people who get it, it's good for them. It's free bonus points. For the others, it's not bonus points. So you'd better actually try to understand what happens, watch the video again and again if needed until you get this right. Choosing an article. Now is a good time. So the deadline is, you still have time, but remember that if you choose an article 1 week before the deadline, you get 2 points instead of 1. So that's 1 bonus point. And it's not huge, but why not? And the reason for that is that if you choose early, it frees a lot of articles for people to choose as well, because then they go in the free pool. So that's to encourage you to kind of be nice with your classmates. If you already know what you want, choose it now, get your free bonus point, and give more options to them. So now is a good time for that, to choose an article. My advice is that you choose an article that you find really interesting. You have the 2 options that are just in front of your name. If one of them really you find it appealing, go for it. If none of them is really your thing and you know what you like, for example, you like bacteria and both of them are about, I don't know, like mammalian cells, then look for the articles that have what you like in the free pool, and if not, have a look at least at the titles for other people, and you can contact them and say like, look, I like your article, are you interested in it, etc. And if not, remember that you can choose really any article you like. It doesn't have to be one of these lists. Like you can go on PubMed, pick an article. I will just make sure that this is not an article that's too difficult for you to write about. And I think the most important is that it's something you like, because writing is difficult. You'll spend some time on it, and if you choose an article that you don't like, you'll see you're not going to enjoy the class very much. One more item on my list: subscribe to the blog. The name of the blog is the Genomic Odyssey. I really advise, if you haven't done so, that you have a look at it. The blog post of January is written by a volunteer. So he is an ex-student of BIOD25, and he loved writing so much for the blog that he did his exam on BIOD25, published his post, and didn't have enough of it, so he wrote another one just because. And so that guy is going to come a little bit later in the course, like after a reading week for sure, and he will explain what he has learned about writing blog posts for the blog. So he will give you practical tips and tricks of where the difficulties are, what mistakes he made, etc. So I advise, go to the blog, see the way it looks, and kind of importantly, subscribe. So why would I ask you to subscribe? It's because many of you will want to have their work published. You know, you will like it at the end of the semester. You think, I did a pretty good job. I'm ready to share it with the rest of the world. Well, you want people to read it. And so if the blog has many subscribers, it will rank higher on Google. So the more you're friendly to other authors now, the more karma will hit you a little bit later, because the blog will be a bit higher in terms of the Google ranking. So my advice is read a little bit, see what the blog posts look like, and kind of subscribe. You know, be nice to them, be nice to yourself. Next item on my list is the login on the Teach cluster. So you will need to have that login set, and you will need to be able to connect to the cluster this week if you want to get max score for the quizzes, because the last question is about connecting to teach. And as long as you can connect, you'll get the points, basically. So on this last question, I just want to make sure that you've done the work of connecting, and then you'll get your 0.1% bonus point if you do that. And no matter what happens, you have 1 week before the practicals, which are in 2 weeks. You have to be ready. You have to have this whole thing set up, because after this we won't help you. We won't help you in the last minute. So you need to do this right now. So I've posted a video on YouTube that explains to you how to do this. In principle, on Mac it's straightforward. I don't expect any difficulties with that. On Windows you have to install an app that's called MobaXterm. It's not very difficult as well. Like I could do it without difficulties. If you have trouble either downloading MobaXterm or getting your credentials, you can contact Michelle. I wrote her email in the previous announcement. Okay, and so of course one other thing that we have to discuss, we will kind of discuss this almost every week now, is return to in-person, because in principle you've seen the memo. You also see that there's contradicting information out there, so it's really not clear what's going to happen. So for us, for BIOD25, it's going to be the week of the 28th of February, the first time when we will start the in-person teaching. For some other classes it could be different, but for BIOD25 it's kind of as late as possible. Right? We have the option to start from February 7th till the 28th, and I have opted for remaining online as long as possible, and the reason is kind of simple, is that we will anyway be busy with connecting to the cluster. There's no reason to do this in class. It's actually more of a hindrance to do that when we're in class. We're much better off doing this from our home to connect to the cluster using your laptops, etc. So there's no purpose for us to be really in class for now, but after these practicals, then it'll make sense. And so I have prepared a poll, and I'd like you to express yourself on whether you want to come to the classes in person, whether you do not want, it's anonymous, huh? Your name in principle will not be shown, whether you will be among the people who come for sure, whether you'll be among the people who do not come for sure, or whether you're like, maybe it's going to depend on the week, so you haven't made up your mind yet. Okay, so I think almost everybody has expressed themselves. So I see on my screen that we have 45% people say yes for sure, like six people say yes for sure, 2 say no, and 6 don't know yet. So share results. In principle, I think you can see on your screens the same results as me. So one thing that's important to notice is that we're 16 people today on the call, on the Zoom call. So that's 15 plus me. So in reality, the class is 40 people. So I don't think that this is a purely random sample of the whole class. The people who are not attending here today, the live class on Zoom, I don't think that the proportions will be the same. I think that we have a huge enrichment for people who first prefer the interactive class, and second, we're also comfortable with, with being present. The people who are not watching this right now, I don't suppose that they will be that much enriched in attending in person. So I'm not going to comment very much on that. That's just for you to think about those numbers, okay? Because I know that some of you are not comfortable with coming to class. Some of you just, it's not handy for them, so it's fine. But for those of you who will make a decision depending on how many people will come, well, like that's 45, let's say around 50% people who say that they're really keen on coming to the class. But that's only 6 people in absolute numbers. Is there any questions regarding that? Things you'd like to discuss while we're at it, because it's an important topic. Anybody wants to, I don't know, get some answer that I may have and you guys don't? I don't see any hands rising. I don't see any question in the chat. So let's get started with the class proper. Okay, so last time was about DNA, so today is really everything about RNA. And here I've showed the structure of the molecules again. And remember that for the DNA we had this 1, 2, 3 phosphate, and then a sugar here, the deoxyribose in the case of DNA, and the base. In the case of RNA, the difference here is on the 2'. Okay, so that's the OH for ribose, so that's the sugar of RNA, and H for deoxyribose. Don't get confused between 3' and 2'. Okay, when you remove the OH in 3', when you remove this oxygen here, remember that you can really not have any elongation. That really blocks everything. But messing up with the 2' doesn't change anything. So that's the difference between RNA and DNA. It's not really that it doesn't change anything. It makes the molecule a little bit more stable. One has got H instead of OH. So RNA has a backbone made of ribose. It contains uracil instead of thymine, and it is mostly single-stranded. These are the very big differences between RNA and DNA. Uracil, remember, is the same as thymine, except that like it's missing this methyl group here. The CH3 that is here in position 5 has been removed from uracil versus thymine. So by the way, it's probably the reverse. It's probably that a methyl group has been added on uracil. Life was probably created from RNA, and at some point there was only RNA. That's what the theory says. And then came proteins, and then came DNA. DNA is the last invention in life, which is kind of ironic when you think about it, given how much emphasis we put on it being the, you know, like the beginning of life, etc., for a person. But on the Earth, DNA probably came later. It's kind of sure by now. And so there was, by the addition of this methyl group on the uracil. So uracil is the ancestor, and thymine is the descendant. Another thing that's quite funny is that DNA was invented multiple times. There are actually two molecules, two enzymes that do this, that catalyze this reaction of adding a CH3 on uracil. So you have really two branches, but they're not even homologous. Like these enzymes are completely different. They have nothing to do with each other, which means that this has happened twice independently in the history of life on Earth. It's got nothing to do with today's lecture, but that's fun. Okay, so let's go back to RNA and let's think about RNA in terms of the eukaryotic cell. So RNA comes in many forms. DNA is kind of always the same thing. Like you could get single-stranded, double-stranded DNA, but it's almost always the same thing. RNA is a different story. You have many variants of RNA. So here that's a sketch of the cell, and you've got different types of RNA which are in the nucleus, and RNA that you will find in the cytoplasm of the cell. So RNA is transcribed in the nucleus. That's where it originates. The only exceptions are viruses, and also transcription in the mitochondria, of course. And then for messenger RNAs, it starts with making a pre-mRNA, pre-messenger RNA. You know, that's the transcript, the primary transcript, that's the way it's called, before splicing. Splicing occurs at the same time as transcription, and then it's exported to the cytoplasm for translation. You also have non-coding RNAs, this non-ncRNA. So the coding RNAs, of course, they code proteins. That's what it means. The non-coding means that they're not translated. That's really the meaning of this non-coding RNA. And the bulk of this non-coding RNA are rRNA, which stands for ribosomal RNA. You also have a lot of tRNA for transfer RNA. And these two things here are, of course, involved in translation. So these are things that you've seen before and that you know. There are a couple of other non-coding RNAs, which are called, for example, short non-coding RNAs. You've got the miRNAs, like the microRNAs, the piRNAs, the siRNAs. So these are very short molecules that are usually interfering with translation or transcription, like miRNAs, piRNAs, and siRNAs. Mi stands for micro, pi stands for peewee. That's just the name of the protein that's involved in this pathway, and Si is short interfering. You also have other kinds of non-coding RNAs, like the long non-coding RNAs. And what are they? Well, they're just long non-coding RNAs. That's pretty much all that we know about them. And they're going to the cytoplasm, but they're not translated. Some of them remain in the nucleus. And then you got pseudogenes. Remember pseudogenes, like they are transcribed quite often, but they don't produce anything that can be translated, or like very, very short pieces of protein. Not very interesting. So So now in terms of abundance, when we look at the mass, okay, like the mass in the cell, so most of the mass consists of RNA, like ribosomal RNA. When you just take the RNA of the cell, something like 80% of it is the stuff that's in the ribosome. That's huge. That's really a huge mass. And remembering that you got 10 times more RNA than DNA in the cell, it means that almost all of it is in the RNA. So it's because you got 10 times more mass of ribosomes than genome. It's kind of funny when you think about it, because when you look at the picture, it doesn't look this way. tRNAs are about 10%. mRNA is the one that actually do the stuff, you know, that code for the stuff, sorry. They're a minority, like 3 to 5%, and the rest is negligible. But now if you look at the number of molecules, tRNA is the winner. Okay, they're not really heavy because they're small, but there's really a lot of them. So like in terms of numbers of molecules, the ratios change, but it's still that you've got RNA and tRNA that dominate everything. So eukaryotic cells contain 10 times more RNA than DNA. Most of it is non-coding, and that's ribosomal and transfer RNA. There are 3 RNA polymerases: RNA Pol1 for RNA, RNA Pol2 for mRNA, and RNA Pol3 for tRNA. So each of them has got their own RNA polymerases. So talking about polymerization, or rather transcription, here's how it works. Yes, there's a question. Go ahead.
Yeah, Dr. Fillion. So I was wondering, like, I know by weight, like mostly it's rRNA, and by numbers mostly tRNA. So does this mean that like rRNAs weigh a lot more than tRNAs?
Yeah, the molecules are much longer. They're really long for or like for the ribosomal RNA. Like you've got these 2 ribosomal subunits. There's the small RNA, the big RNA, and like they really form a really complicated structure. And most of the catalytic activity of the ribosome is RNA. It's not protein. Proteins are here just to maintain the RNA in a certain shape, but really what does the business is RNA. And it's just like one long molecule of RNA that folds itself into a kind of mega machine.
Okay, that makes a lot more sense. Thank you.
Pleasure. tRNAs in comparison are like 70, 100 nucleotides at most. Like they're really short. So Rna synthesis, that's also something that you know, that you've seen multiple times. So when I say Rna synthesis, I speak about mRNA, the messenger RNAs. So you got the process of transcription, starts at the promoter, then the polymerase just goes, elongates the molecule in its 3' because that's what all the polymerases do. It goes like that, and back in the days, people were saying, okay, you've got the primary transcript, and then it's kind of exported to the cytoplasm, and sometime in the middle, somewhere in between, then the introns are cut, and then you got this splicing phenomenon that happens. And when it's somewhere close to the end, you've got the capping, and then it's polyadenylated somewhere in that process. So in reality, it's not really the way it happens. The way it happens is that it's kind of all together at the same time. You got transcription. While you get transcription, as soon as possible, the introns are removed. Like the molecule is not finished, the RNA is not finished, and it's already been spliced in the 5'. That's co-transcriptional. That's really important to understand that, because that's the way that splicing is regulated. So the capping happens as soon as possible. As soon as the 5' end is ready, it can be capped. And of course, you can't add the poly A tail in 3' until you have reached the 3'. So that happens when the RNA polymerase goes to the terminator, and the terminator is not the place where transcription stops. Okay, that's ironic. That's the place where the RNA is cut, and then it grows a poly A tail. But the polymerase goes a little bit further than that. Still called the terminator, because the RNA polymerase will stop shortly after that. So mRNAs are produced by RNA Pol2. Contrary to the textbook view on the left, the mRNA is matured while it is being transcribed on the right. The 5' end is modified with a cap, the introns are spliced, and a poly A tail is added to the 3' end. So this is something you've seen before, but you have to understand that this is all happening at the same time. That's simultaneous. So mRNA maturation consists of capping, polyadenylation, and splicing. You also have some other events like modification of the nucleotides, but we're not going to speak about this. The most important thing for you is to remember that there's a cap, there's a poly-A tail, and the introns in the middle are removed. So mRNAs have no 5' phosphate end. Okay, so when you have usually a single strand molecule, remember that you got 5' phosphate and 3' OH. But it's not the case for mRNAs. What they have is this inverted head-to-head link, which is here with still 3 phosphates that are there. And you can see that this is a 7-methylguanosine. Guanosine is a G. There's a methyl group here in position 7, but that doesn't matter very much. What matters is that it's linked the 5' ends together. So mRNAs have got 2 3' ends and no 5' end. It's kind of funny. Instead, the end is replaced by a modified guanine in opposite orientation. The 3' end of mature transcripts consists of a poly-A tail with up to hundreds of A's. It can go to a few thousands in some cases. So how does that work? You have the poly-A signal here, which is usually AAT, AAA, but not always. Right? That's a consensus. And so you've got other mechanisms. This is just one of the possible sequences which is there. So when the RNA polymerase hits that, it's going to transcribe it. Okay, this is created. And then this is here the mRNA version of the DNA, which is on top. And so it's going to cut the RNA on the next CA if there is a GU-rich tract after that. And so the RNA is cut, and like this A here will be the first of a long series of A that can grow to a couple hundred. That depends a little bit on the cell cycle, plenty of things, etc. Yes, there's a question.
Yes, sir. So I'm just wondering, so like an mRNA molecule, would it still have like a 3' hydroxyl group at the end, since it has like a bunch of adenine?
Yeah, of course. Like it's a normal end. Like there's no protection. Like in the 5', it's just a normal 3' OH. Then there's no particular change at that level.
Oh, okay. Thank you so much.
All right. So it's just a completely normal bunch of A's. Yes, question.
Just out of curiosity, just to I think I know the answer, but I don't remember exactly. Why do we exactly need the poly-A tails? What do we need those long tails for? Just to be sure.
That's a very good question. It's a little bit like think of the mRNA as a bomb, right? So as soon as it's created, then someone will light the wick, and then it's going to go like and it's going to be eaten up.
Yeah.
And as soon as it hits the bomb, then it's going to disintegrate. It's the same. So the RNA, it's kind of a timer. Sorry, like the poly-A tail is kind of a timer that says how long can you live. So there is this, this Greek myth of the fates, and so you have the thread of your life, and there's one that just pulls the thread, another one that cuts it, etc. For the RNA, that's literally like there's this thread which is the amount of life they have, and when they don't have a poly-A tail anymore, it gets destroyed. Like there are enzymes that eats them from the 3'.
And that's for the mRNA, right?
Absolutely.
And the cap, what's the cap for?
It's a very good question. So it's among other things, it's to protect ourselves against viruses. It's a modification that's kind of difficult to make in the cytoplasm, and that's kind of a signature. That's a fingerprint, or if you want a quality control, quality seal that says made in nucleus, if you want. And so, like, if you have an RNA that's coming from a virus, it's going to be hard for them to have that quality seal. And so that's a way to detect transcripts that are not coming from ourselves, if you want. The system is not perfect because some viruses, including SARS-CoV-2, have an enzyme that does exactly that. They kind of stole it at some point, and now they can get away with it. Yes, there's a question.
Yes, sir. So I was just gonna say that I really like your analogy of the bomb. Like, that was a cool one. And so just to clarify, like once the RNA polymerase 2 like goes towards the poly-A tail, that's the signal for degradation of the mRNA. Like, yeah, that's when we stop translating or something.
So the RNA polymerase 2 like starts at the promoter, goes at least until the polyase signal and even a bit further.
Oh, okay.
That's the job's done. But there are other proteins which are not indicated on this slide. One that cuts the RNA, another one that grows the tail, and all this is exported to the cytoplasm and the enzymes that eat the 3' and are in the cytoplasm.
Okay, thank you so much.
Not the business of the Rna polymerase 2.
That makes sense. Thank you.
Right, there's a comment in the chat. I remember that the poly-A tail and the 5' cap also protect from the exonucleases. The exons are protected from digestion. Is that right? Yes, absolutely. So the 5' end, like 5' phosphate, that's a typical attack point for nucleases, exonucleases, especially RNA is very susceptible to exonucleases. So if you did not protect that, then it'd be constantly eaten up there, and that would be a problem. So that's a way, by removing this 5' phosphate, replacing it by a modified 3' OH there, that really protects it from a lot of nucleases that are around. And again, you can think of it, you can even think of the whole transcription system as an antiviral mechanism. The reason why it's so complicated, the reason why it's so messy, is because it's all quality control, and not quality control for errors, but quality control for attacks. It's because we're constantly bombarded with viruses. Every cell of our body has probably been infected at some point, and for sure over 4.5 billion years of the age of the Earth, and at least 3.5 billion years of the age of life. Well, viruses have been around from the beginning. They maybe even were created before cells. I mean, before cells is pretty sure, but even before life, when you think about it. So, like, the reason why it's so complicated is probably because every step is a checkpoint that says, okay, any viruses now will have a harder time imitating that, because in order to do this, you need many genes. Like, there's at least a couple hundred genes that are taking care of transcription in the cell, and for a virus that has a genome that size, it won't be able to have all these genes. But still some manage, as you notice. Okay. So another thing that's quite important in the maturation of Rna splicing or alternative splicing. The process is really complicated, and it's not fully understood at the moment. The actors mostly are understood, that's okay, we got them, but there is a code that hasn't been cracked just yet. So when you give a sequence to someone and say, how is it going to be spliced? Now, most of the time they can't tell, like nobody really can tell. Part of the reason is that it's context dependent. Two different cells with exactly the same sequence will do different things. They will splice at different locations. But even if you remove that and you focus only on the constitutive splicing, it's a problem that's not completely understood. Some people have gotten mutations in their genome, and that changes the splicing code. So it turns an intron into an exon. It's not always understood why. So anyway, the process goes like that. You've got here on top a molecule of RNA with the 5' that has been sequenced, the 3' as well. And so the part that is not sequenced, transcribed, the part here in yellow is the intron that's going to be removed. So there is a U1 SNRP, which is ribonuclear particle, small nuclear ribonuclear particle, U2 SNRPs, which are kind of splicing components that recognize the splice junctions, but again, it's not fully understood how they recognize it. The sequence there is kind of, there's nothing special. There is a GU here, and there is an AG here, but there's two nucleotides on the left, two nucleotides on the right. That happens all the time. So why does it cut exactly here and exactly there? It's not fully understood. Anyway, you assemble a RNA protein complex that makes a loop with your RNA like this, and then there is this attack here of an A nucleotide right at the point of excision, right here. So that makes this kind of lasso here, a lariat structure, and after that the process finishes by cutting the other end here and then stitching together the left and the right. And that's splicing in a nutshell. In reality, people understand how that works, but they don't understand why that works exactly there and there. That's the question. So splicing is the removal of introns from RNA transcripts. They are present in most eukaryotic genes. Most genes have introns. The reaction is carried out by the spliceosome, which sometimes also removes exons. It doesn't only remove introns, it can remove exons as well. By the process of alternative splicing, one gene can produce different mature transcripts. Okay, so when you sometimes remove exons, sometimes not, that's called alternative splicing. And so it just means that you have one gene, one structure, that will produce different RNA. So this is exemplified here. So you've got exon 1, exon 2, exon 3, 4, and 5. Produce always the same kind of like immature RNA or primary transcript, but it will be processed in different ways. So if you have all the exons, it would look like this, 1, 2, 3, 4, 5, so that makes a full protein. But you can also have skipping of exon 3. So then you get 1, 2, 4, 5. And you can have skipping of exon 4, for instance, so that produces different proteins. Okay, so you have questions of reading frame to adjust to make sure that these things don't produce frame shifts. But evolution has optimized all that so that all 3 proteins will be functional. So that's called alternative splicing. So one of the most important reactions at the biotechnological level to understand transcriptomics, RNA, etc., is the reverse transcription. So the central dogma is that genetic information flows from DNA to RNA to proteins. That's the way it was formulated. The dogma is false because reverse transcriptases produce RNA from DNA templates. So the dogma held until the mid or the end 70s when David Baltimore discovered that particular reaction, reverse transcription. Before then, the dogma was formulated by Watson and Crick, and they called it a dogma mostly because Watson didn't know what a dogma was. He thought it was a hypothesis, but like obviously not being a very religious person, he didn't know that. And some people told him like it's not a dogma, that's just a hypothesis. Anyway, so it was false in the sense that it's not true that it always goes from DNA produces RNA produces protein. You have one way in which it can go back, RNA to DNA. And we've seen this in class 1 already with the line 1 retrotransposons. They have this enzyme they can produce from RNA to DNA. So what's this reverse transcription? What is this? So here in blue you've got DNA, that's double-stranded DNA. I highlighted the exons in like in this pale color. So you've got separation of the strands, RNA is indicated in pink, transcription goes like that and produces an RNA, which is here the imaginary primary transcript, which never actually occurs. It's really spliced before it can finish, but let's go with this imaginary transcript. Then the introns are removed, the cap is added here, and the poly-A tail is added. So that's transcription in a nutshell, going from blue DNA to pink RNA. So reverse transcription is the opposite process, of course. So it goes from RNA to DNA. And so reverse transcriptases are from viruses or transposons. We do not really have a reverse transcriptase in our genome. There's only one, but it's a special one. That's the one that puts the telomeres. So example line 1. They produce complementary DNA or cDNA from an RNA template and a DNA primer. The primer can be either poly-T or gene-specific. So because all the mRNA have got this poly-A tail here, you can just use a poly-T primer that will bind the poly-A tail in a reverse complementary fashion. So if that's DNA here, that will bind here at the poly-A tail, or you can also have a primer that recognizes exactly the end of the RNA, let's say there. You put that magic enzyme reverse transcriptase and it will elongate the DNA primer to make a reverse complement of the RNA, so that produces that here. Okay, so that's if the 5' is here, remember that you always elongate from 5' to 3', so that produces the reverse complement of the RNA here, but in DNA form, which means that you got T instead of U inside. Okay, but if you look at it, the introns are gone. Okay, when you go from transcription to reverse transcription, that of course, because during transcription the introns were removed, then during the reverse process they're not there anymore. There's nothing to reverse transcribe. Does it use a DNA polymerase? But by definition it is a DNA polymerase. A reverse transcriptase is a DNA polymerase. It's an RNA dependent DNA polymerase. So why is it a DNA polymerase? Because it produces DNA. It's as simple as that. That's the definition. Okay? So, like the proof is in the pudding for enzymes. An enzyme that produces RNA is called an RNA polymerase. An enzyme that produces DNA is called a DNA polymerase. This one is kind of got a bit of a weird name, reverse transcriptase. You know, you don't really call it transcriptase anyway, you call it an RNA polymerase. So that's kind of a weird way to call it. But it is a DNA polymerase. So Rtpcr, that's a very important reaction that is one of the key components of biotechnologies. So it's the combination of an RT, reverse transcription, and a PCR. And now you've seen both, so things should be a piece of cake. So here you have the RNAs that are coming from the cell. So they're highlighted in pink here with the cap, the poly A tail here, and you first start with the reverse transcription, which uses the T here, the poly T primer that is there. And just as per the previous slide, you go from 5' to 3' and produce a complement here of that particular sequence. So that's the RT. Only one strand is available for the PCR after the RT. Okay, so you produce only one blue molecule here. Okay. At the first cycle, the forward primer can anneal, but not the reverse primer. From the second cycle, both primers can anneal. So I've highlighted the position of the primers here with these ghost arrows. And you can see that as the PCR starts and you use these primers, this one cannot do anything, the one that is really at the bottom. Because there's nothing to anneal to, it anneals to something that is in pink, but the pink is not even present in the PCR reaction. So it's only this primer here that can bind here. It will elongate with the usual polymerase reaction, a PCR reaction. You produce this hybrid molecule here, and then from the cycle 2, then you got 2 templates, and then both primers can start kicking in. Primer sequences are indicated by the outlined arrows. The forward primer has the sequence of the gene. Okay, so that's really is what you read in the gene, the sequence of that primer here. The reverse primer has the sequence of its reverse complement. So that's, if you read the primer from 5' to 3', that's the reverse complement of what you read here. But that's really what I'm saying in the video on YouTube. So if you don't understand what I just said, it means you need to watch that video again. Then of course, if you can do PCR, you can do qPCR, quantitative PCR. And that's a process that many people never heard about in their lifetime until 2020, when of course we know why we need qPCR. And if I had showed something like this 2 years ago, people would say, oh, gross. But of course, by now, I think many of us had to go through this excellent procedure of the swap. Anyway, so the way it works is that you collect the material here, you put this in a lysis buffer that will neutralize the virus and release the RNA of the virus. Of course, here I'm talking about an RNA virus because we know which virus I'm talking about. And you purify the RNA. You do the reverse transcription using either a poly T primer or specific primers in the sequence of the virus that you want to target. And then you make a qPCR after that RNA, like reverse transcriptase. So here that's a high-level description of the genome of SARS-CoV-2. And the way these qPCR tests are done, if any of you had it performed, is that usually you have 2 amplicons which are chosen on the virus here. There's one positive control, which is one of U genes, like an RNA that's present in your nose. That's a human RNA, just to make sure that the reaction works. And then they're going to assay 2 amplicons, the S gene, remember that's the spike, and they will sometimes use the RDRP, the RNA-dependent RNA polymerase, which is something that only the virus has. We don't have it, same as S, or the N gene. So qRT-PCR can be used to quantify the expression of genes, but nowadays it is mostly used to detect and quantify RNA viruses like SARS-CoV-2, of course. So once you're able to do qPCR or real-time PCR, you can also do more interesting things with that. So you kind of increase the complexity as technologies increase. So one thing that you can do, remember that there are 2 systems. There is the cyber system and there is the Tacman system for the quantitative PCR. And the nice thing with the Tacman system is that you can have 2 or 3 amplicons at the same time in the PCR. Why is that? Because you can have a Tacman probe here with a red fluorophore together with a Tacman probe with a green fluorophore. Remember, the Tacman probe emit light when they're destroyed. Like they bind the target, but then the polymerase passes, destroys them, and that's what emits light. And if you do 2 PCRs at the same time in the same tube, if you have 2 independent probes that bind the targets, you're going to emit a red light when this one is produced and a green light when the other one is produced. So you can multiplex the reaction. So that's why nowadays for these SARS-CoV-2 tests, you can test actually the positive control in the human genes, and then the 2 amplicons in the SARS-CoV-2 genome at the same time in the same tube. It's pretty neat. So Rtpcr usually reports the CT value, so that's the cycle threshold. In modern assays, multiple targets are amplified in the same reaction by multiplexing Tacman probes. You cannot do that with Cyber. Cyber is not specific. It will shine green no matter what you amplify. Rtpcr is sensitive, but it sometimes produces false positives or false negatives. So the CT, what is that? You put a threshold in the amplification here, okay, so that's the amount of fluorescence that you can see on the graph, and you say, I put the threshold there, and I'm going to say, when the curve hits that threshold, that's the point I'm going to report. If I hit the threshold early, it means there was a lot of material to start with. If I hit that threshold super late, it means there was not a lot of material. I needed to amplify it a lot more before I could detect it, so there was not a lot. So people just report the CT. And the reason why I'm bothering you with these details is because sometimes nowadays when you are positive with the SARS-CoV-2, they give you the CT. They say CT45 or CT35 or CT30. And so empirically, if you have a CT which is after 40, it comes from only 1 molecule in the tube. Like if you had 1 RNA molecule, that's enough to give you a detectable level after 40 cycles of PCR. So it means, yeah, okay, you may be positive, but if this molecule is in the air, if you just fell in the tube or something like this, then you'd get the same result. If you get a CD of 30, this is a lot more. That's 1,000 times more. Okay? And so depending on which CT you get, you can suspect that's a false positive that's coming from contaminations in the air, or no, that's for sure, like this person's got a lot of virus. So of course, once you can do RT, reverse transcription, and you can do PCR on it, then you can also sequence it, of course, and that's the whole point of transcriptomics, genomics of gene expression. And the method is it's pretty much what you would expect, basically, after what you've seen. So you have a sample, a biological sample, you isolate the RNA here. After this, you do an RT, so you produce cDNAs in DNA form, you generate cDNA fragments, you select the size, and then you put this on a Illumina sequencer. So that's the glass slide that we've seen last time. You can see the starry night with the stars of different colors in the background. If you remember, that's the videos that we saw last time about the Illumina sequencing process. And then you can do paired-end sequencing on these RNAs. You don't really sequence the RNA, you can't. You sequence the cDNAs. We can't really sequence RNA with Illumina because we need a polymerase that amplifies DNA. So you can only sequence cDNAs. And after that, you got reads that are here, so they're indicated with the red and the black ends. So black is left, red is right. And if you map them to the genome, you will see that they map only to exons, of course. Like you can't really sequence introns. They have been removed during transcription, and the cDNA hasn't added them. And so you will see reads that map to this exon, but that leave the introns alone. They don't map there. So you can kind of know how the gene was spliced because you see which introns have been removed. And then you can do some downstream analyses, of course. That's just getting to know which are the genes that are expressed. So one difficulty, what people don't really understand in general, is that you cannot just put DNA in the sequencer and sequence it. Okay, you've seen last time, this video is pretty intense, the one with the Rpkm, etc. That really shows that in order to sequence something, you need to pre-process it quite a lot. You need to have known ends on the 5' and the 3'. If you don't have these known ends, you won't be able to sequence it. And so there are different ways to produce something that you can sequence from RNA, because just throwing the RNA on the sequencer is not going to work. The most popular methods are template switching on the left and RNA ligation on the right. So I'm briefly going to go through the process. So again, we've got these RNA that are here, so 5' to 3' spliced cap polyadenylation. You take a poly-T primer, and remember that when you do PCR, the same for RT actually, you can modify the 5' end. You can put whatever you want on the 5' end. It's not going to interfere with the priming. You're not allowed to mess up with the 3' end, but you can really put whatever you want on the 5' end. So you can put already on the 5' end whatever you need to be able to sequence it on an Illumina. So you do the RT here, and then that produces the cDNA in blue, and you take a special RT, special transcriptase that adds a couple of Cs here at the end, and it does this for free. They're called untemplated Cs. So it adds 2, 3, sometimes 4 Cs here, and that's enough to use another primer that has only 3 or 4 Gs, and that will allow to prime here that reaction. So you'll band there, and then it will elongate like that. And then you've got, if you have put again on the 5' end, whatever you need here for sequencing, you're done. It's a pretty neat trick, because you have relatively simply added something you need for sequencing on the right hand and something you need for sequencing on the left hand, and you're good to go. You can sequence them. The other method is to ligate these pieces that you need for the sequencing. So you randomly break the RNA into small pieces, and then you use a ligation, like a ligase, for RNA. So it really takes the 3' end of an RNA molecule and really makes a covalent link with a 5' end of another. So you ligate special adapters that have a blocked 3' end, so that they don't form concatenamers. The only thing that can happen if you block that is that this primer will find one of the RNA molecules that you've generated by cutting randomly here at that stage. After this, you've got only one free 5' end, which is there, and you can use another adapter that you can ligate with the same method. And so that will ligate whatever you want here, whatever you want there, and then you can just put primers to do a PCR to amplify that. So again, these are things that you don't need to worry about when you're actually not working in the sequencing center or not actually doing the operations. You know, in the Hollywood movies, they just collect the DNA and they sequence it directly. Yeah, right. There's a lot of processing that needs to be done before you can do that, because in order to sequence something, you need to know a part of the sequence. That's a pure chicken and egg scenario. So you need to put these sequences there so that you can start the sequencing. It's only because everything depends on a primer. You need a primer, you need to have some piece of the sequence that is known. It needs to find its way in your molecules. It has to be the case. Little exercise, stop and think, why can we use the right protocol on total RNA? Okay, so if you do this on total RNA, like you extract all the RNA of the cell, what's going to be the problem? Why can we use the left protocol? So I extract all the RNA of the cell, and people ask me, Guillaume, which protocol should I use? I say, the left one. This one is, you're going to get a big problem if you do that. What is that big problem? Okay, so I think we're going to make a little break here because it's 3, and then I'm going to go back to these previous slides, but have a little break, like 3 to 5 minutes, and then we come back together at 6 past to 8 past, something like this. Okay, so now we're going to go to the next topic, which is quantification, and that's a topic that's relatively important because it's not completely trivial how to quantify the expression of a gene. So there are 2 methods, like 2 units that people use very widely in order to quantify Rna-Seq, and the first is called Rpkm, and Rpkm stands for reads per kilobase per transcript per million map reads. It is a unit of transcript expression that scales by transcript length. Okay, so it's kind of a complicated thing. So let's read it again. Reads per kilobase of transcript per million mapped reads. So when the transcript is long, then the Rpkm increases in a sense. Well, actually, you'll see it kind of decreases in reality. So why is that the case? It's because if the transcript is long, then it's got more chance of capturing reads. Okay, if the transcript is 1 kb or 2 kb, the transcript of 2 kbs will naturally have twice as many reads just because it's got more surface. Okay, so if they're expressed at the same level for one molecule of each, one will produce twice as many reads just because it's longer. So that's something that's really important to remember. So here's an example of how to quantify this. Gene 1 has got 0.5 million reads, and they look like this. These are the blue little thing here. When there's a thin line, it means that the read was split. It was not really mapping on the intron here because you cannot sequence introns that have been removed. So it's kind of spanning over the intron. So it's just telling you that this has been removed by transcription and hasn't been put back by the reverse transcription. So you got the gene is tiled like that. You got 1 kb worth of exon length that has been tiled by blue reads like that. Gene 2 has got 1 million reads, so twice as many reads as gene 1. Okay, but when you look at it, let's say it's 2 kb's worth of exon, the tiling is about the same density as gene 1. Okay, so if the tiling is the same density, intuitively you'd like to say they have the same expression. Even though that guy has got twice as many reads as gene 1, it's not because it's twice higher, the expression. It's just because it's twice longer, and it's those reads here that contribute to multiplying the number of reads by 2. It's not the fact that you got more density over the same length. Okay, so how do you do that? So you compute the total number of reads, which is 1.5 million. Okay, so 0.5 million plus 1 million. And you say this guy here has got 0.5 million divided by 1 kb divided by 1.5 million. That's the reads per kb, so 0.5 million reads per kb per million map reads. So you divide again by the total, which is 1.5 million, and you get an Rpkm of 0.33 million. The other one, you do the same operation and you get the same number here. Okay, so you do 1 million reads divided by 2 kb, so that gives you 0.5 again, and you divide by the same number, which is 1.5 million. So both of them have got the same expression, and that's the purpose of this unit. Okay, we want to say that if the gene is a bit longer or shorter, that should not matter for the expression. What should matter is the number of reads, of concentration of reads per unit length of the transcript. Sorry, could you clarify what a read is again, please? Yeah, indeed. So if you've been here in the last class, you remember that we're using, for example, Illumina sequencing to do high-throughput sequencing. So when we talk of Rna-Seq, it means that we're just using Illumina or any high-throughput sequencing in order to directly sequence the transcriptome of the cell. Okay, so that here, every read is one thing that has been decoded in your high-throughput sequencer. Any idea how many reads you get in a typical experiment these days when you use a high-throughput sequencer for Rna-Seq? The order is approximately, 500 million. That's what you have to remember. 500 million reads is what you'd get for one experiment. Okay, so in practice it's more like 300 million, something like that, 2 times, because you got the left hand and the right hand. But that's what I mean by a read. Like you got, you put your DNA in the machine, your cDNA in the machine, and then you get a file which has 500 million entries, and these are what has been decoded. Those are the reads. Yes, Theodore.
So okay, so we're looking over. We're saying this is the 1 million reads, 0.5 million reads. Now my question is, like, if we're like, what could be like an example, like a simple dumbed down example of scenario where we would be using this kind of thing? If it's okay to ask, like,
I mean, I don't think there is a simple dumbed down example where you would use Rna-Seq. This is a pretty advanced technology already, and explaining Rna-Seq to your grandmother or something like this is going to be hard anyway. So I can't think of anything right now. There are many cases where you'd you know, where you'd use units that scale with length or something like that. But like, if it's the sense of your question, like, why would we scale by length? Then let me find, is there an example where we could do the same? Right. So, you know, when you compute the GDP of a state, you'd say, okay, like the GDP of China is bigger than the GDP of the US. I'd say, really? Is that the case, that Chinese are richer than the US? You say, well, not really. If you divide by the number of people, then you say, okay, how much money is there per person now? Well, then it's much less. And of course, it's kind of the same idea here. If you want to measure how rich is a transcript, how many reads does it have? You know, like, it's not a very good analogy, because now it's the length of the transcript, it's not the number of people, but pretty much everything you have for any kind of unit, you need to normalize it with how many contributors are there to that unit. So if the gene is twice longer, it will contribute twice as many reads, not because it's expressed more, but just because it's twice longer. I can't find a much better analogy right now, but maybe at the end of the class we can discuss that. For now we need to make progress, and there's another question. Sorry, the calculation, why do we add 0.5 million plus 1 million? Is it because both genes are on the same transcript? No. This is the total number of reads. Let's say it's a tiny genome with two genes. That's a really dummy example here. In general, you will have 20,000 genes, and counting the alternative transcripts, it will more be like 100,000. But let's assume that we have a tiny virus genome with only 2 genes. So it looked like that. So we do this because it's the total for the genome, not because they are on the same transcript. So it's per million mapped reads. If everything we mapped is either in gene 1 or in gene 2, then the total number of mapped reads is 1.5 million. Okay, and that's why we divide by 1.5 million. And then we multiply again by a million because we want per million mapped reads. That's why we get 0.33. If you're confused about the calculations, don't worry. You just need to write it down, review that. It's going to take you 2 or 3 minutes, not more, at the end of the class. It's just that during the class, it's hard to follow detailed calculations. But when you pause for a second and you do this, you'll see it's pretty super simple. So Rpkm is the number of reads in a transcript times 1,000 divided by the exon length times 1 million. So that gives you that. Divided by the number of map reads. Sorry, that gives you that. So it's not a simple unit in any way. It does the job in the sense that both of these have got the same Rpkm, and that's good because intuitively we want them to have the same expression. They look the same. The read density, like the evidence of expression, is the same for both reads, for both genes, sorry. But the problem is that 0.33 kind of is a bit of a weird number, and it's completely arbitrary and doesn't really tell you anything nice. So we'd like to have another unit that has this property that gives us something kind of a little bit less arbitrary than why 0.33, why not 0.7695? Where does that come from? There was never a 0.33 anywhere in that example. So this is why people came up with the other score, which is called TPM. That's much more popular at the moment. TPM stands for transcripts per million. It is a unit of transcript expression that scales by the transcript length. So it's the same as the Rpkm. It is the number of reads in a transcript divided by its length, scaled so that the sum is 1 million. So here, if you don't understand what that means, it's okay. I don't understand it either when I read it. I just put it there for reference. what you need to understand is that you take the Rpkm from the previous slide and you say, I want the total to be 1. So I'm just going to give them a coefficient so that now the sum is equal to 1. And well, obviously it has to be 0.5 and 0.5 because they're both expressed at the same level. And that is more intuitive because that says it's 50-50. 50% of the reads go here, 50% of the transcripts are coming from that gene, 50% of the transcripts are coming from that gene. So when you look at the cell and you take a transcript at random, you got a 50-50 chance that it's going to be gene 1 or gene 2. And that's finally something that's much more intuitive. So Rpkm, people don't use it anymore very much because it's not intuitive at all. It's okay because it does the job of making sure that long transcripts don't get more expression just because they're long, but it's not intuitive. TPM, on the other hand, really gives you a probability distribution over the transcript. It tells you if I take a read at random, what's the chance it's that gene? And that's what we need. So one difficulty as well is quantifying splicing, and that's kind of adding some difficulties. The score that people use is PSI, person spliced in. It is a measure of the inclusion of each exon during splicing. If the exon is excluded, spliced out, its PSI value is 0. If it's included, spliced in, its PSI value is 1. Intermediate values indicate intermediate alternative splicing. So you look at the evidence of reads that are not including exon 2, these are the reads in red here, and you look at the evidence of reads that are including exon 2. These are the reads in yellow. And you can see that if you do the math, you'll say I got so many % of the reads that tell me that there is exon 2. That's for sure because I sequenced it. I mean, you never sequence introns, of course. And so many % that tell me that there is no exon 2. And the proof that it is not there is that they really start with exon 1 and they continue directly in exon 3. They really are evidence that exon 2 is not expressed. So you do the sum and you see that, for example, in this case, it's 80% of the transcript must have exon 2. So that's a PSI of 80%. Okay, the person spliced in is 80%. But the scenarios can rapidly become complicated because if you have multiple exons, multiple transcripts, etc., it can rapidly become difficult to understand how to quantify this property. So I'm not going to bother you very much with that. I just want you to know that it exists and that it can be a bit difficult to compute in practice. But the idea intuitively is this one. How many % of the transcripts have got that exon? That's PSI. So I've got some more promotional videos. Today we have 2 of these short promotional videos. I hope you're not sick of them. And after this, you're free of promotional videos for the rest of the class, I promise, for the rest of BIOD25. So one thing that I'd like to talk about now is single-cell Rna-Seq. And that's one of the, of the most important developments of the last decades. And that's why I wanted to show you that. This one doesn't say anything, so there's no need for the sound. And this is taken from a company that's called 10x Genomics. They are the leader in single-cell transcriptomics. Right, so they have this machine here, so it's like a press button machine. This is the method here. So you put the cells in that tube, then you put the tube in the machine, you press enter and you're done. And then you sequence it. So that's why it's so popular. Okay, now how does it work in practice? They have these gems. Gel bead in emulsion partition. And so the point is that they have beads here with DNA on them. We're going to see in a second what that is. There's this cartridge here, put that in the machine, and it does everything by itself. But of course it costs an arm and a leg. These things are really expensive. Okay, now pay attention. You've got here the beads, and then you're throwing cells on them, and then you've got these cells that are in these tiny emulsions. So each emulsion here, each bubble has its own reaction. We're going to see this in a second. You produce the Illumina sequencing library in these tiny bubbles, and after this you can sequence everything. Alright, so you got for each cell the expression of each gene. And after this you got a beautiful picture. Okay, make every cell count, we got it. Okay, so the principle, we're going to see this in a bit more detail right now, is that it's called emulsion PCR. You do everything in these tiny beads. So that's the picture that they showed in the promotional video, and that's using microfluidics. So they have this tiny, tiny tube in which they can fuse things together. So you've got oil here at the end of the tube, and what they have here is water. Okay, and they're kind of bombarding the cells on these beads. The beads, I'm going to describe what they are exactly in a minute. For now, just imagine them as colored things. The cell sticks to the bead, and then this is thrust in oil. And so at that stage here, you've got an oil ocean, and you've got tiny bubbles of water in that ocean. And many of these bubbles contain a cell, and they contain only one cell in that little bubble. And the point, the genius of this method is that in that bubble, the cell is isolated from every other cell. So they're going to do the whole Rtpcr, no kidding, the whole Rtpcr in that bubble. And the bubble is never going to break during the time when they do the Rtpcr, which means that every cell gets its own Rtpcr. And if you can do that, it means that after, if you just sequence it, you're going to get fantastic stuff out of it. As long as you can tell, these Rtpcr products are not the same as these Rtpcr products. So let's see how they can do that. The beads here are coated with primers, and the primers are different between every bead. The primers are the same. You have many molecules here. These are these kind of wiggly, like hair if you want, on the bead. These are just primers, DNA primers, and these primers are all the same for the same bead, and they're different for different beads. Okay, so these primers contain a poly T tail, so that should ring a bell. That should say, oh, that's how the RT is going to start. So it's with the poly T tail here, and they have a UMI, which is a unique molecule identifier. So that's a sequence that's different for every single primer in the world. And a 10x barcode in green here, that's the one that gives the color of the bead. On a bead, they all have exactly the same 10x barcode. So now if you do the Rtpcr reaction with these primers here, it means that the cell which is on that bead, all the reads will have this green part here, the 10x barcode. So when you will sequence a little bit later in the Illumina machine and you find this, this particular sequence, the one that's in green here, you say all the transcripts that I'm seeing with that green sequence, they come from the same cell. I don't know what cell it was, but I know that they all come from the same cell. So scRNA-seq method, single cell RNA-seq methods, gives an overview of which genes are expressed together in the same cell at the same time. So how does that work? Is because you can sequence that index, and that's something that we have skimmed over very fast last week, is that there is the P5, P7 adapters, the sequencing primers for the Illumina technology, but you also have the index. And that's something that we very briefly showed last week. Like here, outside the molecule, on the flanks, you're allowed to sequence the index. And so the way it works is that instead of sequencing forward in the, in the amplicon here in the Illumina sequencing reaction, you take a reverse complement of that sequence and you sequence backward. And that's the one that is here, I7 index primer. And that's sequencing that little green part that was here. And that's how you can actually decode these so-called 10x barcode that were here during the reaction. It's all based on that. So the Illumina workflow reads an index I7 after read 1. The index is placed at the position where it does not interfere with anything, and it can be sequenced before the cluster is folded over. So you sequence it before read 2, before the reverse read. The commercial name is TrueSeq, in case you want to Google it. That will explain to you how you can sequence these indices here. Indexing allows multiplexing different samples in the same lane. So that's one way in which you can put multiple samples together. But in the case of single cell sequencing, you're putting multiple cells together. Okay, each cell has got its own index. So you don't have only 10 or 16, you have now millions of these indices. And you can then easily tell which are the reads that come from which cell. And so you make then packets of cells. And so you can use projection methods in which each dot is a cell and all nearby dots have similar transcriptomes. So at the end you get a very big table that says one row is a cell and one column is a gene. You got 20,000 genes, maybe 1 million cells in the experiment, if you pay a lot of money. It makes you a very, very big table that says each cell has got all these genes expressed at these levels. But what do you do with that? You can use methods like PCA or TSNE, in which you project that in 2 dimensions. And now you represent one dot as one cell, and 2 dots are close when the transcriptomes are similar, and 2 dots are far when the transcriptomes are not so similar. For instance, when you do that with a method that's called PCA, you can see that the cells form clusters. And what these clusters are mostly G1, G2M, and S. It's kind of obvious that cells at different parts of the cell cycle express different genes. They express the division genes or not the division genes. And so when you just look at a high level which are the genes that are expressed in the cell, mostly what you find is that these are the genes that differentiate every cell is where they are in the cell cycle. Are they resting? Are they dividing? Are they close to mitosis? Are they replicating their genome? So there's a lot of genes that need to be turned on for every of these conditions, and that's why they will form different clusters. If you have a complex organ like blood or something like this, then you will see other kind of groups. Like it could be CD4 T cells, B cells, CD8 T cells. Like the groups will correspond to different things, just based on the identity of the cells, not necessarily the cell cycle. But typically you would use this kind of method to tell you, I had a mix of different cells. Now, what are the cells that look like each other? What are the cells that don't look like each other? And you use these kind of techniques. Another very important trend that's developing at the moment is called spatial transcriptomics. It's how to get the transcription of every cell, not really based on the cell, but based on where it is in an organ. So you have, for example, this tissue section of the brain here, and you'd like to know which are the genes that are expressed in different areas of that organ. For a kind of uniform organ like the liver, is not very interesting because there's only one mass, but for the brain or for breast, something like this, or the heart is some organ that's pretty complex, then being able to do spatial transcriptomics allows you to tell you some sub-organs of the organ, to say here it's expressing these genes because that is happening, and there it's other kind of processes happening that express different genes. Also think that you can have, for instance, a cancer, and so being able to tell these are the cancer cells, I can see them there because they express completely different genes, and these are the non-cancer cells with a normal expression, that can be quite useful. So you take a tissue section and then you use this kind of method that I'm going to show you in a second with the last promotional video that says, okay, these are little pixels here, and each of these pixels has got its own transcriptional profile. Spatial transcriptomics designates methods to map transcription in tissues. That's again the 10x Genomics company. They are the leaders in these things at the moment. And the technology is called Visium. So you got a slide like this where you can put your samples. So you can put up to 4 different samples. And then they use their technology, the one that they have developed previously. It's all based on bar coding. And so instead of barcoding cells, they will barcode pixels in the sample. All right, so here everything is based on whatever on these cells. So they've got different barcodes that are now on this 2D matrix. Like locally, it's the same primers that are like on different pixels of that grid. And these are the same primers we've seen them before. So you put your sample here. And so the cells will leak their RNA locally only. So like every piece here is a pixel with different primers, and they leak the RNA, but only locally. So they're captured by different primers that are here, and they have different barcodes, different indexes. Once you have made the cDNA, it's fine. You can just dump everything together. They each have received a mark that will allow to differentiate them. Okay, so the point is that after that has happened, they have leaked locally the RNA. You're using the same technique of indexing to be able to say these RNA are coming from the top left corner, these ones are coming from the 1 pixel on the right, these ones 1 pixel on the right, etc. So that makes you a grid in space, in 2D, of your organ. So why do we care about gene expression? So one thing is that we want to know something about gene regulatory networks. So only a fraction of the genes in the cell are active at any point. There's not really any cell in the genome that expresses all the genes. So each cell expresses only a specialized part of the transcriptome of the genome. So what determines which genes are on and which are off? Something has to take decisions to say brain cells will express brain genes, and then liver cells will express liver genes. Who decides that? Some genes regulate other genes. It's their job. This induces networks of genes activating each other, which in turn activates effective genes that specify the cell type. Example, B cell or T cell, and what it does. Example, divide, produce antibodies, cytokines, etc. So here that's a overly complicated representation of a transcriptome, but that's pretty much how it is. Like you have one gene that activates another gene, that activates another, that activates another, and that feeds back on the first. And then you got a chicken and egg scenario. So that creates these feedback loops where when you start to express that genes, then you're going to maintain that particular expression. And two cells will have they will explore different areas of that space, and they would express different genes. But that's all based on these so-called gene regulatory networks. The idea that some genes, their only job, they're not producing anything. They're the bosses. You know, they're telling who should be expressed and who should not, but they're not doing any of the job. The ones that are doing the job are usually at the end, at the bottom of these hierarchies, and they would actually produce the things, like produce the cytokines, produce the antibodies, make stuff for real. But then there is, at the top of the pyramid, these genes that decide what are we going to do in this cell. Is that going to be an antibody, a cytokine, or is that going to be something for neurons? And so these genes, most of them are transcription factors. Transcription factors are proteins that specifically interact with DNA. They usually bind the major groove and recognize particular sequences, but not with Watson-Crick type of interaction. So here you have a zinc finger representation. So that's a classical type of transcription factor. And this is the way it decodes the sequence in the DNA. Like it turns around the DNA molecule like this, and each zinc finger recognizes 3 nucleotides in a row. So with 3 zinc fingers, you can recognize up to 9 nucleotides kind of turning around the DNA molecule like that. And as you can see, it's not reading the DNA by the Watson-Crick base pairing. It's kind of going on the side, recognize the base pair really from the other atoms. It's not really bothering at all the binding of DNA with a typical Watson-Crick base pairing. It gets the information, but it doesn't interfere with the annealing of DNA. So you got another representation here of that particular binding, and you can see that you recognize the phosphates backbone here. They're there in red, and the contacts are exquisite. Like this particular protein here will have one atom at exactly the right place so that it can make a hydrogen bond, for instance, with the phosphate group that's there, and another one with the other phosphate group that's there. But the binding with the phosphate is not doing anything for recognizing a specific sequence because all the DNA has got these phosphate groups here. But you got some of them that have a loop here that goes inside the groove that will make specific contact only when you got the right nucleotide, A, T, C, or G. Here you've got a zoom in just to show you how exquisite that can be. Like you got a tyrosine that's here. That particular tyrosine has got an OH here that will really recognize one of the atoms of the either C, A, or T that is here in the DNA. So that's the level of specificity. You got some atoms that are interacting with a backbone, with a phosphate backbone, some others that really go inside the groove to kind of decode the sequence. So they usually facilitate transcription by recruiting components of the RNA Pol2 machinery, but their mode of action is still poorly understood. Even though they're totally fundamental, it's still kind of hard to know exactly how they work. So one important part of the process is regulatory sequences, like these pieces where we need to have transcription factors that come recognize them in order to activate transcription. And most of them are enhancers. And these are sequences that activate transcription at a distance. We've seen them before. They are bound by transcription factors, and they are believed to loop over to promoters and help recruit the RNA Pol2 machinery. So the enhancer is here, and you will see in the model in the textbooks that there's this model where it loops like that so that it can touch the promoter. And this is based on plenty of experimental evidence that there needs to be a proximity between enhancers and promoters. This model is still in the textbook and it will remain in the textbook for a long time, for decades I suppose, but it's probably false. So here's a primer of what's probably true instead, but this is still in the making, so this is like research in progress, and that's why it will probably take a long time before you see this in the textbook and before your teachers tell you that's the way it works, that's the norm. So the most recent model is that enhancers help form microenvironments where transcription is easy. Those microenvironments are short-lived, they live minutes, seconds, minutes is really at most, and they're formed randomly. You know, this is like supernova explosion after this disappears, but that supernova explosion will allow transcription locally to happen. This would explain why transcription comes in bursts and why bursts are synchronized for two genes that are nearby. This also explains why genes change expression if they are transplanted to other locus of the genome. This model is probably correct, but the details of the mechanisms are still unknown. So how does that work? You can think about it with this here. So there's kind of a mini bubble of transcription that will happen, which will take several genes in it. Like, yeah, think of it as an explosion. Like there's something that happens here that changes locally the concentration of the molecules in the nucleus, but very, very locally. And that allows transcription factors to really come in as if it was a black hole. That really attracts all the transcription factors there. And once that happens, transcription can proceed extremely easily, but only for a short time. After this, like a black hole, it will kind of explode, release everything, and transcription will stop. But for this, let's say 10 seconds to 1 minute, all the genes that are inside that black hole, they will be transcribed super heavily at the same time. So that these three genes that are there, they'll fire everything they've got for 1 minute, and after this, silent. And so that's what people see experimentally now using the techniques that I've showed before, like Rna-Seq and single cell, etc. You can totally see this, that transcription comes in bursts, 1 minute of transcription, and after this, 3 hours of nothing. And that these bursts are exactly synchronized for two genes that are close on the chromosome. But if the genes are far apart, transcription is like firing unsynchronized. Also, that explains why you change the place of a gene, then it will synchronize with another gene that's close by, but not with the previous genes that were there. So these are kind of the type of evidence that shows that this is correct, but there's much, much, much more that shows that probably you have these microenvironments that really permit transcription, and that explains what we've seen before. But it would take a long, long time to explain all of it. That's it. That's all I wanted to share with you today. So the key points to remember are that RNA is a diverse molecule and it's involved in many functions. mRNA, messenger RNAs, are a minor fraction of the total RNA in the cell. It's almost nothing, like 5%. And regulation of transcription is a complex process. We've got these beautiful technologies to study it, but it still remains a horribly complex process that is mostly not understood yet. So now it's time for breakout groups. So we have 2 exercises for today, and as soon as I'm done guiding you through the exercises, I will dispatch you in the breakout groups. So exercise 1 is the following. You have a little sketch that's here. It says that the reporter on the left, so here that's a reporter, that's a piece of DNA, is introduced in a neuron. The reporter consists of the line 1 retrotransposon, we've seen it before, the line 1 retrotransposon, plus the reverse complement of the GFP, interrupted by an intron in the forward direction. Okay, you can see GFP here, and there's intron that goes like this, that goes forward. The neuron becomes green fluorescent. Explain what happened. What does this say about the retrotransposon? Okay, so try to see a little bit the features of that stuff. Think about transcription, cDNA, reverse transcription, etc. And try to understand why when you put this here in that neuron, then the neuron would turn green. How would that happen? What has happened? Exercise 2, immunosequencing. Immunosequencing is a method to monitor the diversity of the immune repertoire, B cells and T cells. It is used to diagnose lymphomas or immunodeficiencies. Both are characterized by a loss of immune diversity. So you've got one antibody that goes like sky high. Design primers for immunosequencing on an Illumina Rna-Seq. You must obtain a PCR product that can successfully go through the bridge amplification. So the P5 and P7 oligos attached to the glass slides are this and that. Okay, so here the V part is indicated with the sequence that is here. The J part is the sequence that is in orange. And so you want to sequence what is in NDN here. Okay, so you need to design 2 primers that are there, and these primers must be able to work on an Illumina sequencer. So think about primer design. That's the burden of this exercise. Design a primer pair that will allow you to just do directly a PCR on the genome, and then you can throw that in the sequencer. It's gonna work. All right, so I'm going to stop the recording now. So if you're watching this on the recording, then see you next week. For the other guys, we stay here.